Okay, I think that we're recording and we're kind of good to go. So let me just move a few things around and we'll make a start. So what I had on plan for today was really for us to have a look of kind of a condensed version of your weekly material for this week. So this week we've been looking of a review of basic concepts in chemistry of life, cells, and also a little bit about the genetics. All of this should be very familiar material for you from when you took the classes that were required as a prerequisite for this course. So I don't expect that there's going to be anything hugely shocking or surprising, but I still want to offer this chance to kind of have a really condensed version of what we've been looking at this week. So as the very first thing, I was going to do a check-in and just see how everyone's doing and kind of touch base where I can help you even better. So even though this is an asynchronous class, it doesn't mean that you're stuck doing things by yourself. I also wanted to remind you of what's to come for this week, what I'm expecting to get as submissions, as pieces of homework from you. And I was going to quickly review the content of the chapters 2 to 4, which we are looking at this week, really based on the areas that students most feel that they have questions on. There's no way that we can review everything, but just kind of touch upon any tricky areas. Since it's me here by myself today, and it's going to be probably more of a recording that students check out, I'm going to pick out a few areas that I think that are most important. But if anyone wants to later on have any specific areas addressed, we do have our weekly meeting also available tomorrow night. So that's an option for you. If we have time, I was planning to kind of quickly touch upon on the homework so you have an idea again of what to expect with that. So when I think of how we're doing, I always think that it's really important for me to know what's going on in our students' life. You don't have to disclose specifics, but if your life has a lot going on, it might be harder to focus on what you're supposed to be doing in a class. So I want to remind you that we have a lot of resources available for you as learners through the institution. So we have various services that might be of a help if you ever feel that that help would be of a use for you. I also think it's important that we talk a little bit about kind of regularly checking in what's working well in this class. And also, where can we do better? From my perspective, since I'm the only one, I'm going to ask myself tonight or this afternoon that what's working well. I'm very impressed seeing that you guys are doing a great job getting assignments submitted in on time. And the assignments that I'm getting back are of a high quality, which tells me that you're definitely grasping the material. I do think that we might revisit the times and days for these weekly live online sessions if they remain very low in attendance. These are not required by any means. I offer them as an additional resource, but if it turns out that I'm alone a lot on these, then I probably will reconsider that how many of these sessions do we need. I also think that it's super useful every once in a while to also kind of trust to the universe, that universe guides us to where we need to be and what's going on. So I normally do with my students an activity from an interesting book by Young Pueblo, which is titled Inward. So we take a thought of the day and kind of touch upon that. But I shall not do it by myself because that's really probably limited in terms of reflections that we might get out of that. But let's see if we have more attendees tomorrow, we might visit that. Like I said, these live sessions, I offer 2 per week. The content of the both sessions is the same, and I will also record both sessions. So we have the one today on Wednesday afternoon, and I will offer one again tomorrow night on Thursday, starting at 8 o'clock. And like I said, I will record them, I will make them available, and there's absolutely no pressure. I know that many of us have other commitments, and these courses are designed to be asynchronous, so you don't need to be joining at any particular time. But I do offer that option for those students who feel that that's beneficial to have that chance for direct exchange. Do remember that I also offer live online office hours in a virtual setting, Mondays, Tuesdays, and Wednesdays from 1 to 3. And links to those are available on your Canvas course calendar. So I encourage you to use that as well. This week, what I'm really looking for you to do is actually just 2 quizzes. There's no practice anatomy assignments. There's just simply 2 quizzes. And the quizzes that I'm asking you to complete cover chapter 2, and then the second quiz covers chapter 3. Both of these are due at the end of the week, so 1 minute before the end of the week on Sunday night, and both are worth of the same amount of points. They're completed on Connect. You will be able to access it through our Canvas course. I also wanted to quickly kind of highlight what to expect for the next week, because next week is going to be a little busier. Next week, we're completing 2 chapters. So we're completing chapter 6 and chapter 7. In addition to the chapter quizzes, which I have one for each of those chapters, and in addition to the chapter practice anatomies, I also have our first food for thought assignment. It's more of a kind of a chance for you to apply what you've learned, chance for you to write a little, very, very short answer, maybe a paragraph in length, let's say maybe 5 sentences. It can be whatever you think is sufficient for you to deliver the answer to the question, but that's lined up for the next week. In addition, next week, you will be taking your first of 3 exams. So the first exam covers chapters 1, 6, and 7. And in addition to that, you'll be taking also your first practical test covering the very same chapters. Both the homework and the exams are due at the end of the week next week. So keep an eye of that. With all that having been said, I think that we're in a good place for us to have a look of some of the key points of the material that you would be covering this week. So let's do that as well. And just let me get my screen oriented. So your first chapter this week really kind of took you back to considering the basic chemistry that's the requirement for life to happen. So really chemistry is kind of fundamental foundation for all of the other things that you'll be covering in anatomy and physiology, because chemistry really explains the small stuff. And so we can start building these bigger systems of the body. Chemistry explains the composition, the structure, also the behavior, properties, reactions that can take place with other substances for atomic, molecular, and ionic level of materials. One thing that I kind of thought that would be interesting to remind you is that 96% of your body is made by 4 main elements. So your body is largely made of carbon, hydrogen, nitrogen, and oxygen. In addition to that, we have a broad range of other elements, but these elements together combined represent about 96% of all of your body composition. The first concept that I wanted to talk about real quickly is the concept of matter. And when we think about what matter has really two basic qualities. Matter has a mass, and matter takes up space in the universe. So what I mean by mass is often in students thinking, you can think of that mass is similar or interchangeably used to a weight of an object. But remember that mass is the absolute unit, whereas weight is dependent on the gravity. So the mass is constant regardless of what gravity you're on, whereas weight is dependent. Your weight will be very different on Earth than what it would be on the moon. Sometimes my students tell that, oh, they're not they're not heavy or this or that, they were just born on wrong planet. If that helps you to think about it, please feel free to do so. What I mean by the fact that matter takes up space, one way to consider it is that we know that this marker that I have on hand, it does have a volume. It takes up space in the universe. Therefore, it must be made of matter. So those are the two qualities that we can use to define what matter is. Other things to kind of consider is that similarly to matter having a mass and taking up space, matter can sometimes change the form that it is in. So in case of water, for example, water can take solid, liquid, and vapor form, gaseous form. Obviously, water doesn't have a plasma form, but that's the fourth form that is feasible for certain elements, but not all. And that brings me to talk a little bit about the elements. Elements are pure substances that cannot really be broken down into further substances by normal chemical means. It turns out that some of the elements that we have discovered, we are now able to break even further, but early on and in normal setting, most elements are broken down as far as we can break them down. What I mean by that is that it takes a lot of energy, it takes a lot of effort, it might take a very specific tools to break elements that we know currently as to any further. So really elements are the fundamental materials that all of the matter that we have is composed of. And that brings me to talk about the diagram that's at the bottom of this screen, the diagram of a periodic table of elements. Periodic table of elements is a way to illustrate all the known elements that we are aware of at the moment. And we're organizing them in a very specific manner by grouping them based on various characteristics, and the way they are grouped on this periodic table of element might be very helpful for us when we're trying to predict how different elements behave, either alone or together with others. There are 92 elements and a periodic table of elements at the moment, that naturally exist 26 that we can further acquire by doing further work in the lab setting. But this periodic table of elements has been revised multiple times, so it is not unheard of a thought that that might change as well. And I provided you a periodic table of elements as an additional handout on your Canvas course for this week's material, so you'll be able to download or print it from there for your own use. Now that we're talking about elements, sometimes students ask me that, well, what is the relationship of an element to an atom then? By large, I do see those being used interchangeably, even though it's a slightly different tone that we're using when we're talking about those. So atoms are really the smallest unit of ordinary matters, and they form the chemical elements that we're seeing. And what we're going to do next, we're going to have a look of a typical atom. So atoms vary greatly, but most atoms contain few key characteristics, and I wanted to quickly visit on those. So atoms typically are made of a nucleus, and within the nucleus we have our protons and neutrons. electrons. So these are all subatomic particles, particles that the atoms are made of. Protons are positively charged particles, whereas neutrons have no charge. So those are at the very center of the atom. And around this nucleus of an atom, we have electrons that circle around it. And these electrons, you can consider them almost as like tiny little planets that go around and around that nucleus on certain shells of typically of a certain distance, same distance away from the nucleus at all times. The electrons carry a negative charge. So often we provide you an illustration such as the one that you see here when we're trying to explain what an atom is. I do want to make a caveat here to point out that these illustrations are simplifications, that in reality the atoms are much more dynamic and much more changing, especially the electrons are not following just a two-dimensional flat sphere around or plane around the nucleus. They tend to rotate much more three-dimensionally. So keep that on mind when you see these diagrams, that life is more complex than the basics that we use to explain life. I also wanted to tie these atoms to the periodic table of elements, and here we're seeing one element from the periodic table of elements, and typically with the abbreviation of that element we can also find other information from this labeling that we have on a periodic table of elements. So when we are looking at the atomic symbol, typically on top of it you will also find the atomic number of that particular atom, and at the bottom of that you would find the atomic mass of that particular element. So atomic mass is calculated by adding together the mass of proton and mass of neutron, and that gives us the mass number for that particular element. The mass of electrons is really rather small, so it doesn't have a significant contribution to this. So the convention is that we just try to keep it simple by focusing on the mass of proton and neutron. Other thing that I wanted to point out here was, in terms of the atomic mass, is that we might have variations of a chemical element that have different number of neutrons. Typically the proton number remains the same, and that would then obviously change the atomic mass for that particular element. We call these variations of a chemical element in terms of the number of neutrons as isotopes of that element. And these isotopes give us a rise to the concept of radioisotopes. So these atoms that contain unstable combinations of neutrons and protons or have excess energy in their nucleus can be harnessed for purposes such as medical imaging as tracing agents, or they can be used for aging materials, figuring out how old organic materials are by analyzing the decay of the radioisotopes. They can of course be also used for harmful purposes. We sometimes have seen those in case of nuclear weapons and so on. So let's jump back to our periodic table of elements, and here I've listed it and I've shown you that periodic table of elements was used to arrange all of these known elements in a very particular manner. We will see periods which are running kind of vertically as the columns of the periodic table of element, and then we have groups that run as rows horizontally on the periodic table of elements. So sometimes if you hear people referring to a certain period or certain group of a periodic table of element, now you have an idea of what they're referring to. I also wanted to say a couple of words about molecules. The term molecule refers to when we have two or more atoms that are held together by different kinds of chemical bonds. And because of that, these molecules are typically electrically neutral. By that, I mean that the charges in this molecule typically end up canceling each other out. When we talk about electrically neutral atom rather than a molecule, the concept is the same. So we're seeing the same number of electrons as there are protons. Remember, electrons carry the negative charge, so we should have as many protons with positive charge for that electrical charges of an atom to be neutral in total. I also wanted to mention real quickly the concept of these electron shells. So remember, these were the planes around which the electrons circled the nucleus of an atom. And there's multiple shells, and we always feel these shells in an electron starting from the innermost one. But what's noteworthy is that there's a limit of how many electrons can we fit into each shell. The innermost shell typically can contain only maximum of 2 electrons. The second shell that we see can hold typically maximum of 8 electrons, and then we move onwards and onwards and onwards with the atoms that have more electron shells than that. The atom, obviously, then what's important for us is to know how many electrons it has, and especially how many electrons it has on its outermost shell. The reason why we focus on that outermost shell is that if we are taking away electrons from an atom or we're adding new electrons to an atom, it typically happens on this outermost shell where the changes first happen. So a lot of us focus on what happens on this outermost shell, which is called valence shell of an atom. If the valence shell of an atom is full, so it contains the maximum number of electrons that can fit in to this outermost shell, this valence shell of an atom, we describe that atom or that element as being stable. So it's not, the stable elements, stable atoms are not very reactive. They're kind of happy where they're at, and they don't really search to change that. They've reached that state of having the full outermost shell. However, we also have sometimes atoms that only need to lose one or few, or only need to gain one or few electrons on their outermost valence shell. And if they do so, then they will have a full shell. So these electrons that are so close of getting to this full electron shell, full valence shell, they are known to be very reactive. So this kind of elements are eager to jump into various different reactions because they're trying to reach that state of a full electron shell for the valence shell. So in this illustration that I have here, we're seeing we have 7 electrons on a valence shell. So it would need to gain only one more to get a full valence shell. So it's going to be very, very active. It's just one short. So it's a very reactive substance that we're seeing here being illustrated. Conversely, we could have a substance such as the one that we see on this screen that has only one electron on its outermost valence shell. The shell and underneath or before that, so in this case the second electron shell is actually full. It has 8 electrons. So if this element, if this atom finds a way to get rid of that one electron, it would reach a perfect spot of having a full valence shell. So again, these kinds of atoms, these kinds of elements are very, very reactive and very likely to join in various different reactions that we see. So again, by losing that one electron of that previous example that we had, now we would have an atom that has reached a stable state. It has a full valence shell. So the valence shell that we're seeing here contains now 8 electrons on the second shell, so that makes it full. Now, this relates to the periodic table of elements. So in periodic table of elements, we can look at, based on the number of electrons on the atom's valence shell, that how reactive or stable that specific substance is. So we'll see that there's a group of very reactive elements that we see, and there's also a group of very stable elements. These stable elements had the full valence shell, whereas these reactive ones either needed to lose one or gain one. They were very, very close of getting to that stable state. So we can simply, by looking at the location of an element on a periodic table of elements, predict how reactive it is going to be or how stable it is going to be. This really leads us to talk about chemical reactions and what's happening in chemical reactions. So like we saw earlier on, one of the things that we can do is just simply get rid of electrons to reach that full valence shell, or we can also have a situation where we need to just gain one or few electrons to get that full valence shell. So the big goal is really to get that full valence shell, full outermost layer, outermost shell of electrons for that particular element. So this really explains why and how we see chemical reactions. Chemical reactions are these processes where we're rearranging what is in that atom or in that molecule, and we might move parts of the atoms around in order to get as many of these atoms to the state where they are in a state that they're striving towards having that full valence shell. So these original substances in chemical reactions are known as reactants, whereas these products, these new substances that we produce, are known as products. So reactants are basically what goes into the chemical reaction, and products are what comes out. I have a couple of examples of that. Let's have a look at those quickly. So here we have 2 hydrogen atoms, 1 oxygen atom. And if we were to put those into reaction as a reactants, they would rearrange themselves to form H2O, so our water molecule. So rather than being individual atoms, these atoms can join together to form a molecule, and in this case, it would be a water molecule. So water would be the product, reactants would be those 2 hydrogens and 1 oxygen going into that reaction for each water molecule that's being produced. We can have other kinds of reactions. Here we're seeing hydrogen peroxide over time naturally tends to break apart into water, 2 water molecules and 1 oxygen molecule. So that's why if you buy hydrogen peroxide in a jar and you leave it to a room environment and you don't close that jar and you leave it for a long time, all you're going to be ending up with is water and oxygen. These kinds of reactions where, such as here, where the hydrogen peroxide breaks apart into water and oxygen, and that's the only direction where the reaction can go, they are known as irreversible reactions. So we can't take water and oxygen and just in a normal setting, room setting, just expect them to form hydrogen peroxide. It doesn't work that way. The movement is only to one direction in this reaction. It's known as an irreversible reaction. We do have some reactions where the reaction can move back and forth to both directions. So for example, here the reactants being bicarbonate and hydrogen, which can then give rise to carbonic acid. Similarly, carbonic acid can actually be converted into our bicarbonate and hydrogen. So which one is product, which one is reactant can take turns. These kind of reactions are known as reversible reactions. They can go both ways. And the truth of the matter is that a lot of the time in life we see that these reactions can be multi-stepped. So it's not just one reactant that turns into a product. There might be multiple middle steps, such as here where we see that bicarbonated hydrogen can be converted into carbonic acid, which can be converted into carbon dioxide and water. But similarly, carbon dioxide and water can be converted into carbonic acid, which can be then converted into bicarbonate and hydrogen. So these are longer of reaction chains. And this is really important when we look at the way, for example, how pharmacology works. We don't have to impact every step. We just need to find one step in a reaction where we are able to make an impact. So all of this that we've discussed has been to kind of prepare us to think about another topic or another concept, different kinds of bonds that different elements can form. And the first of these bonds that we're going to be looking at is known as covalent bond. I often describe covalent bonds to my students as a statement that sharing is caring. So in covalent bonds, these different atoms that form a molecule end up sharing some of their electrons. So let's have a look of that. So here we have two hydrogen atoms, one oxygen atom. You notice that how hydrogens have only one electron on their valence shell. So in an ideal situation, they would gain another electron and they would now have a full first valence shell, first electron cell that they have would be full. It would have reached that state or that they were striving towards. Oxygen atom instead has now 4, 5, 6 electrons on its outermost shell. So in an ideal situation, that oxygen could gain 2 electrons and it would reach this full valence shell, which was its ultimate goal. Well, how do we solve this situation, making sure that everyone is going to be happy? Well, how we're going to do that is by having these elements, these atoms sharing some of their electrons. So you'll see how these two hydrogens share their electrons with the oxygen. And even though no one fully owns these shared electrons, so they're kind of sometimes with the hydrogen, sometimes they're with oxygen, a lot of the time both of these elements, both of these atoms are happy. So really it's about finding a situation where we're able to fulfill those goals of everyone, even if not all the time, at least most of the time, by sharing these electrons. And often what we see is that we draw these shared electrons in chemical shorthand or shorthand in chemistry as a line. So a single line symbolizes a pair of shared electrons. So that's what I'm showing there. And it's not always that these electrons are shared in a fully kind of a diagrammatically nice plane. Sometimes they might be crooked, sometimes they might be in an angle, as we see here, for example, in how water molecule was formed. And we'll talk about water molecule and why do we find this kind of an angle in the shape of a water molecule a little later. But what you need to take away from this is that that pair of shared electrons is typically written in shorthand as a single line. We can, of course, have situations where we share two pairs of two shared electrons. In that case, we would draw two lines. So again, that chemical shorthand will tell you about those electrons, how many are shared and between what elements of the molecule that's being formed. And whenever we're sharing these electrons, whenever these atoms are joining together, they are forming a bond. They're tying together, they're forming a molecule. So they're no longer just floating around by themselves. We have situations where these electrons are equally shared between the two atoms that form that molecule. So these electrons spend just as much time with that first oxygen as they spend with the second oxygen. So in these cases where the electrons are shared equally, we talk about non-polar covalent bonds. So there's no polarity between these electrons. They are equally shared. However, we can have situations where we actually share these electrons in an uneven amount, so that the electrons spend, let's say, for example, in this case, a little bit longer at one end of the molecule than then spend in another end of the molecule. So there would be an uneven amount of time that these shared electrons spend with the hydrogen versus what amount of time that they spend with the oxygen. So this would cause these molecules to have polar covalent bonds. Polar referring to that unequal sharing of these electrons. And that might be very, very important later on when we look at how different molecules might behave. So with all that having been said, I want to introduce you, since we're going through the key concepts quickly, to second kind of bond that we find, which is known as our ionic bonds. In ionic bonds, there is actually a net gain or lose, so the actual atom actually completely loses or completely gains some electron or electrons. Here I have an example of that. We're seeing, first of all, an substance where we have only one electron on the valence shell. So in an ideal situation, it would try to get rid of that one additional electron that it has on its valence shell, so that the remaining shells that are left, the second shell, would be full. As we see there with the 8 electrons. The other example that we see there is has 7 electrons on its valence shell, so it would very gladly receive 1 electron to reach that full valence cell, and then it would reach that stable condition. So what can happen, as it happens in these mixtures, is that one atom simply gives away electron, another atom simply gains an electron, or it can even be more than 1 electron. When we do so, we have to remember that we had those charges. Electrons were negatively charged. Now the atom can either become overall slightly positively charged or slightly negatively charged, depending on whether it gained or lost that additional electron. So it doesn't have any more neutral state. There's a slightly positive or slightly negative charge when we add all the charges together for the particular atom, for the particular element in our reaction. So these positively overall charged substances, so these substances that have lost an electron, so they now have slightly positive charge, they are known as cations, whereas these substances, these atoms that have gained additional electrons, they have a slightly negative charge now, so they're known as anions. So really, these ionic bonds that we are seeing here are based on the fact that opposites attract to each other, so cations are attracted to anions. If we have substances, say, as we see often in salts, that there's both positively and negatively charged atoms, they tend to form these bonds, and these bonds were known as ionic bonds because negatives and positives attract each other. So that's how they stay together in ionic bonds. The final one that I wanted to introduce to you of the different bond types was the hydrogen bonds. Hydrogen bonds are kind of covered as a separate group simply because water is so essential for life. So water has some really important characteristics. So we're going to cover that as its own type of a bond because that's really so fundamental and so foundational type of a molecule that we have decided to call it the attraction of water molecules to other water molecules as hydrogen bonds. So what causes hydrogen bonds is that really when we look at the molecular structure of a water, it's made of 2 hydrogens, 1 oxygen. There's an uneven sharing of those shared electrons between the hydrogens and oxygen. So we remember that this uneven sharing of electrons was called as a polar covalent bond. So really, I guess you could say that hydrogen bonds are a form of a covalent bond, but they're just a very specific type. The foundation is in a form of a covalent bond, and that gives us a rise for further considerations. So in this case, now we're seeing the single water molecule that the electrons spend significantly more time, these shared electrons, with the oxygen end of this atom than they spend at the hydrogen end of this atom. So for that reason, because the shared electrons that we are carrying a negative charge spend longer with the oxygen end of this atom, water molecule, becomes negatively charged, whereas the hydrogen ends of the water molecule get to spend less time with those electrons, so they become overall slightly positively charged. So we are left with this diagrammatic illustration of a water molecule where the oxygen end was negatively charged, hydrogen end positively charged. These charges are very, very minute, but they exist. Now we can just simply draw that water molecule as having those charges, and if we put it in an environment where there's multiple other water molecules around it, we notice that there's a spontaneous arrangement of these water molecules based on the opposites attracting each other when it comes to these charges. So there's weak hydrogen bonds between positively charged ends of one water molecule to other water molecules negatively charged ends. So this is how water molecules are attracted to other water molecules, and these hydrogen bonds are what we're seeing illustrated here. I do want to make a note that these are very weak bonds between the water molecules. So those were our three types of bonds that we saw, and what I wanted to really highlight next was couple of key properties of water, why water is so essential for the chemistry of life. And a lot of that, again, I expect is revision, but we'll have a look of that quickly. So we know that water is extremely good solvent. A lot of things dissolve into water thanks to this polar nature of water molecules. So water is largely used as an excellent solvent for a lot of things. We also see that water has a cohesive characteristic. So because water molecules are attracted to other water molecules, that gives specific properties for water. For example, an example of cohesion where water molecules are attracted to other water molecules is seen in this illustration where we have a glass of water, and you can actually fill the glass of water even higher than its rim because these water molecules are still sticking to other water molecules. They don't break apart. We can actually form that kind of a dome-shaped surface thanks to the cohesive forces of the water on that glass that we're seeing. Adhesion instead refers to the attraction of water molecules to other surfaces, and these other surfaces are especially polar surfaces. So we see this, for example, and we see little droplets of humidity on surface of the plants in the mornings and so on. This is also explanation why we're seeing capillary action. So when we have very, very narrow, small tubes, water tends to sneak up on those small tubes, even higher than where the initial water level was. So capillary action is being used largely by many plants to transport water as well. Kind of building up on that cohesive forces of water, we see that water also has a high surface tension. Certain bugs, for example, utilize this in terms of walking on water, but we see that as well in other implications. So, for example, when a plane has to do an emergency landing on water is not always the best option. Water surface, if you approach it in a wrong angle, can be really hard, just as hard as a concrete. So you might have seen an example of that if you've done any swim jumping. So you jump from a high elevation to a, let's say, lake or sea or pool. And it really matters in which angle you break that surface of the water. If you have done a belly flop, you've noticed that you have this large surface area that's hitting the water at the same time. It's hard to break that surface tension, and you will really feel it and probably even develop some bruising. Whereas if you're able to break the surface tension of water with a small sharp angle, such as your toes or fingers going and breaking that surface first, that might not be as a hard surface that you're facing. Another characteristic of water is that water is excellent at storing heat and slowly releasing the heat. This is why we see that a lot of people prefer to live on coastal areas, because during the hot days, the big masses of water are storing the heat, and during the cold nights, these big masses of water are slowly releasing the heat. So in this way, we're able to kind of balance off those extreme temperatures and kind of get more consistent environment. Obviously, here in Arizona where we live, we do not have high amounts of humidity in the air, so we see really big changes between nighttime and nighttime and daytime temperatures, because there's not that water or big bodies of water to store that heat and release that heat slowly. We of course remember that water can take different forms. Water can take a liquid, solid, or vapor form, depending on in which environment we're at. So that might also be worthwhile to notice about the water. And this kind of different forms has further subsequent consequences, some of which we will visit in just a few minutes. Finally, like I mentioned, because water is a great solvent, it kind of ties into the fact that we're able to transport a lot of materials also with the water. So water is a great way of moving things around, both in animal body, as we see, for example, with the blood. So a lot of things are moved in the blood, and blood was mainly made of water, but also in the plants, we're able to transport a lot of materials with the water. So it's not just the water that the plants extract through the root system from the soil. With that water, also nutrients travel to the tissues of those plants. So that's another example how water is a great transport medium. That was a lot to cover. Let's really quickly talk about certain things, certain materials being attracted to water. These are known as hydrophilic substances, anything that's attracted to water, whereas hydrophobic substances tend to avoid water. They don't want to get into a direct contact with the water. Well, why does this matter? We see this, for example, when we see here an illustration of a cell membrane, plasma membrane, which is made of phospholipids. Phospholipids have a hydrophilic and hydrophobic end. So the hydrophilic ends are facing towards liquid, whereas the hydrophobic tails are facing towards other hydrophobic tails. So they're staying as far away from the water as possible. That's the explanation of the spontaneous arrangement of the phospholipids to form these cell membranes. The question that I wanted to ask from you relating to the properties of water, the different forms that water can take, is that why does ice float? Well, the explanation is actually rather simple once we think about it. In a liquid form of water, these water molecules that make up the water mass are much more tightly packed together. So they're denser for that same unit area or unit volume. Whereas on ice, water molecules need to take this kind of a crystal shape arrangement, and this shape arrangement pushes these water molecules further apart from each other. So actually the same amount of water when it's frozen takes a larger volume than a same amount of water when it's liquid. So we see that ice really is lighter because the same amount is spread on a larger volume. So this explains why water tends to expand when it freezes. And we've all seen examples of that. You shouldn't put sodas in the freezer if you're not willing to take the risk that if they freeze so fully, the bottles might explode, and so on and so on. We see examples of that when people have frost nights and they leave water into the garden hoses, and then the water, as it freezes, expands and tears those hoses apart, and so on. So really, this is the explanation why ice floats. So the liquid water is very dense, whereas solid water taking the ice solid form, it becomes lighter because the molecules were further apart. One more topic that I wanted to talk about real quick was to do with the pH scale. You remember pH, the H in it tells that we're measuring the concentration of hydrogen ions. So it's really a mathematical representation of how much hydrogen we find in a substance. Running from 0 to 14, pH 7 being neutral, anything below 7 being acidic, so containing lots of hydrogen ions, whereas basic substances containing little hydrogen ions, so anything above 7. And we can classify many different materials based on where they fall on pH scale. We of course have buffers in the body which kind of tend to neutralize these changes, pH changes, as needed. So that kind of brings us close to wrapping up some of the chemistry. I did want to mention real quickly about the concept of organic versus inorganic chemistry. So organic chemistry often is considered as looking at materials that are of a living origin, whereas inorganic ones are typically of non-living origin. But another way, if we look at the chemical structure of organic materials that are organic in nature, is that these contain typically carbon and hydrogen. And this carbon is particularly important because carbon has 4 electrons on its valence shell, making it particularly reactive. So many organic materials are very, very reactive thanks to the carbon backbone of these materials. So we talk about carbon or hydrocarbon backbone of many organic materials, and that really explains why we are seeing these organic materials being very, very reactive. I also wanted to show how in organic chemistry, we see that materials can arrange this hydrocarbon backbone in many, many manner, even though the chemical structure of a material would be the same. So on these diagrams, we see 6 carbon atoms forming the basis or the backbone of the molecule that you're seeing. So same number of carbons, but very different shapes. So that means that all of these are different isomers of uh, of a same substance, same molecule. So there's just a different arrangement of materials. Another thing that you sometimes see when you talk about the chemistry and chemistry of life is that we have various different kinds of functional groups. These functional groups are parts of a molecule that have specific properties, specific characteristics in terms of their chemical behavior. They always behave in the same way. And again, I provided this list of key functional groups to you in your materials as an additional handout. Really quickly, I kind of just wanted to remind you that molecules were made of 2 or more atoms, and we find certain molecules in nature they're the most common molecules that we find. So we talk about essential molecules that we typically find, and these are going to be carbohydrates, lipids, proteins, and nucleic acids. Let's go through these really quickly, and we'll learn a little bit more chemistry as we go through them. So before we go through them, I just want to point out the concept that when we talk about monomers, mono meaning one unit that molecule is made of, whereas polymers are made of multiple units that have joined together. So poly for many. How these monomers are made into polymers or polymers are made into monomers, well, let's have a look at that process on a theoretical level. So here we're seeing multiple individual monomers on the top line. So these are not connected, they're just individual monomers that are hanging about at the same area. If they now decide to join together, what happens is that they form a large polymer as a result of dehydration synthesis. So monomers are joining together, and in that process where they're joining together and rearranging the chemical structure from individual monomers to one larger polymer, they're also releasing water as a byproduct. So that's where the name dehydration comes for, and synthesis referring to making something new, in this case, like making a larger polymer. The opposite reaction can also take place where we have a large polymer that's broken apart into individual monomers with the help of adding water to this chemical structure. This kind of a reaction is known as a hydrolysis. We use water to break apart, to lose a polymer into monomers. We do see in nature reactions where this can go back and forth as well. It's not stuck one way or another. For example, salt water, we can help to evaporate the water and salt remains behind, but we can also dissolve salt into water. So this reaction can move either way if we want to see. Or sugar cubes are a good example of how we can dissolve sugar, this kind of solid material, into water, and then again it will crystallize if we remove the water from there. This brings me to talk about the concept of enzyme. Enzymes in general, you can consider them as things that assist in chemical reactions, and how they assist, that can vary quite a bit, whether it's making it easier, faster, more efficient, quicker, all sorts of ways, lesser activation energy. Enzymes just help a reaction to happen that would happen otherwise, and they help by bringing that activation energy, that point where that reaction happens, lower, as you can see in that diagram that I've illustrated there. Another way to think of an enzyme is really to think what it looks like on a molecular level. So here the yellow parts are the enzyme, the green part is some sort of a substance that the only goal that we have is to break it apart into two parts. And this reaction would happen normally, but by adding the enzyme, it might happen now faster or more effectively or more completely or with lesser energy. So we're really seeing how, while acting, enzyme might change its shape, but once it is finished doing its job, it returns back to its original shape. In addition, enzymes can be reused time and time again. So enzymes are not used away as they are helping with chemical reactions. A lot of textbooks mention catalyst as an example of an enzyme. Catalysts speed up chemical reactions. That's what these type of enzymes do. So not all enzymes are catalysts. Catalyst is simply one example of enzymes, but probably the one that most often gets talked about. So coming back to this listing of carbs, lipids, proteins, and nucleic acids as important biological macromolecules, let's have a look of carbs. So when I talk about carbs to my students often say things like, where sources of carbs, let's think of bread, potato, corn, rice, pasta, all that good stuff. But there are other sources of carbs as well. A lot of leafy greens, for example, vegetables are good sources of carbs. Carbs are used in the body as raw materials and fueling, giving us energy to do many processes. So I assume that this is really where our kind of natural inbuilt tendency and attraction for consuming carbs comes from the era when food was much more scarce. So carbs taste good because they contain a lot of energy, a lot of resources for our body. So we are kind of naturally attracted to those. In addition, I wanted to mention a few words about fiber. Fiber is non-digestible components. They serve a few different purposes. First of all, fiber gives us kind of the bulk so that we feel full, but also helps to move materials along the digestive system. So consuming some amount of fiber, even though there's no really actual energy or raw materials in it, is considered as something that's good for your digestive processes. If we are looking at different types of carbs, you remember we talk about monomers and polymers. In terms of the carbs, we see saccharides. We see monosaccharides, really simple single molecule sugars, for example. We can see disaccharides, a little more complex, made of 2 units of saccharides, 2 glucose units. For example, lactose in the milk and so on. And then we have very, very complex carbs, such as polysaccharides, such as we find that in, let's say, whole wheat bread. There's very, very complex polysaccharides. So this really simply tells us that how complex the carbohydrate structure is. And we typically end up finding that those monosaccharides are easier used in the body as a source of energy than polysaccharides. But sometimes it might be good to have a very complex carbs because they last around in the body for longer. I also wanted to mention quickly chitin. These are polysaccharides that give exoskeleton. So a lot of bugs don't have a skeleton, internal skeleton such as us. Their skeleton is actually on the outside of their body, such as in case of cockroaches. You can consider this exoskeleton almost as a medieval kind of armor that's kind of keeping the animal's body together. They don't have those internal bones, so the bones are on outside. So I sometimes think about a lot of these bug sprays that work by dissolving the chitin, that what a horrible way to go when your bones dissolve. Like I mentioned, complex carbs are generally considered as better sources of energy than simple carbs. That's because the simple carbs are available for body to use as a source of energy so quickly, whereas these complex carbs remain around for much longer, so they don't get burned off as quick. So that was our carbs. Let's look at lipids real quickly. Lipids, when I talk about those to students think of things like butter, oil, lard, cheese, things like that, ice cream. But there are also other sources of lipids that are considered more healthy, such as fish, nuts, seeds, avocado, olive oil, things like that. Lipids are really used as a highly concentrated long-term form of storing energy. Again, I think that that's why we are attracted to eat foods that contain high amounts of lipids, because our body evolutionarily has been like, oh, this is a great way of getting a lot of energy. Lipids also play an important role in the fact that they can form, because lipids are insoluble in water, they can form these protective barriers. Whether we're talking about leafy surfaces, waxy surfaces of the leaf, or we're talking about the fur of an otter, for example, that repels the water from getting through the fur. Lipids can be very useful in that sense. Let's have a look at lipids a little bit more. In this case, triglycerides. Triglycerides are made of 1 glycerol and 3 fatty acid chains that are attached to that. And again, as we learned earlier on, of course, we can break these triglycerides apart through the process of hydrolysis into a separate glycerol and 3 fatty acid chains, or we can join these separate glycerols and 3 fatty acid chains together during dehydration synthesis into 1 triglyceride. So let's have a look of a single fatty acid in a little bit more detail, and not all fatty acids are alike. We have saturated fatty acids where we see single bonds between these carbons. These saturated fatty acids are typically fats that are solid in room temperatures. They are associated with a higher risk of cardiovascular disease, whereas unsaturated fatty acids have at least one, if not more, double bonds here and there, and this double bond causes kind of a kink in the structure of that fatty acid chain. These unsaturated fatty acids are typically liquid in room temperatures, and they are considered as a healthier source for human consumption. So they're the better option. In addition to talking about saturated and unsaturated fatty acids, since we know that saturated fatty acids are not great for us, but they might be a little bit more tasty or attractive for some, humans actually did come up with an artificial way of doing these unsaturated fatty acids and trying to convert them to have more characteristics like saturated fatty acids. And this is what we're seeing when we're talking about hydrogenated fats or trans fats. So this is done through an industrial process where we add hydrogen to liquid vegetable oils to make them solids. And this causes these hydrogens on opposite ends of this double bond to take a shape. And there are some concerns whether hydrogenated fats are really great for you, especially when it comes to the bad cholesterol that might be associated. I often talk to my students about manufacturing process of margarine, but I'm not going to go into more detail here on that. But if you're interested, you can pause and have a look of it is quite a wild process how margarine is being manufactured. So that might make you want to think about that again. And really, if we're thinking of these lipids, that's the explanation of how cell membranes are made. Cell membranes are made of multiple phospholipids that spontaneously arrange into two layers with multiple phospholipids side by side. So that's where the name of a cell membrane as a phospholipid bilayer, so two layers, comes from. Here we're seeing a single phospholipid having been enlarged at the bottom corner of the picture from that cell membrane that's at the right upper corner. This phospholipid has a hydrophilic head, so it's really attracted to the water. It's happy to be around the water, whereas the tails were hydrophobic, tried to avoid water. So that's why the tails tend to spontaneously pack facing against each other, so they get as far away from water as possible. When we talk about lipids, we should probably also mention steroids. These are four-ring structures arranged in a specific manner, often where hormones might be produced and so on from the cholesterol and so on. In terms of the proteins, I wanted to say a few words, even though I didn't have an opening slide for that. I must have deleted it. Proteins, good sources of proteins, egg whites, beans, tofu, meat, milk, things like that. Proteins typically are not stored in the body, and proteins are usually used only as when no other source of energy is available. So we go for these other sources first, carbs and lipids. This is why it's so hard to lose weight. Proteins are important for building muscle mass. If there's no proteins available, or no other source of energy available, your body will start burning off its own muscles as a source of energy as well. Proteins are used for many different purposes. So we'll keep seeing different kinds of proteins time and time again in A&P. They provide support functions, metabolism, transport, defense, regulation, motion, but also structural components of the muscles, for example. Proteins are, in a chemical sense, polymers of amino acids. So they are made of a structure where we have this variable part. And again, I've listed these different kinds of combinations that you can find as a handout that's on your materials. With proteins, it's important to note that we can look at proteins on many different levels of complexity as a simple amino acid chain or a flat sheet-like structure that bends, or more three-dimensional structure. So it gets more and more complex as we study proteins and how protein takes shape. We can see that, for example, in how red blood cells take shape, and if there's errors in the coding on that, we end up with sickle cell disease. So that was our proteins, and with the proteins, like I said, the ideal order how your body tries to use these different different macromolecules as a source of energy is that carbs are preferred, then lipids, and only then proteins. This is where these low carbs or no carb diets are based on, that if there's no carbs available for energy, your body would start to burn off lipids, so fats, for a source of energy. So that's the idea behind those. Finally, a few words about nucleic acids. So nucleic acids, the ones that you know are DNA and RNA, DNA storing genetic material, whereas RNA being important for converting that information into a protein structure. Nucleic acid has a general structure where we have sugar that has a phosphate group and then a variable base group that could be adenine, guanine, cytosine, or thymine in DNA, and we change thymine to uracil in RNA. We can also study body in many different levels of organization, cell being the smallest level of organization, the smallest unit. Cells are generally small. There are some cells that can be long. For example, we see in a spinal cord, spinal neurons can be up to 1 meter of a length, but by large, cells are small. In terms of studying cells, we have different tools, light microscopy available for us, electron microscopy available in other facilities that allows us to go even to smaller material. So we can see cells just barely with a naked eye, but none of the cellular structures really. It's the light microscope that we need to see the nucleus, see the cell walls. And if we want to see those smaller cell structures, we really ought to use electron microscopy for that. Like I said, cells are small because by biological design, it's ideal. So we want to keep the surface area to volume ratio as optimal as possible so that we don't use the materials that we're trying to transport across or into the cell just because the distances are so great. If we're studying cells, the field of studying cells is cytology. The field of studying multiple cells that form tissues is known as histology. First, sometimes I get students asking me that how did the first cells come about? Cells have all this stuff inside, and based on our best understanding, it is because first cells started as a symbiosis of materials that by accident ended up inside the others that were to become a cell. Cells can be divided largely into two groups, the prokaryotes and eukaryotes. Prokaryotes were the first to evolve. They're smaller, simpler structures. They have a free DNA floating inside. There's no really membrane-formed separate structures. Whereas eukaryotes were later to evolve. These are what we see, for example, animal cells. They're larger in scale. They have specific areas of cell that are specialized for different purposes. So the compartmentalization of the cell is very real there. And DNA is housed inside a membrane-enclosed nucleus. So really the key when we're studying about animal cells for our purposes is to pay attention to this compartmentalized nature of eukaryotic cells. So the first structure that I wanted to mention was the plasma membrane of the cells, like I said, made of two layers of phospholipids that we just saw, so often referred as a phospholipid bilayer. So referring to the fact that it's made of these phospholipids. Phospholipid term comes from the fact that they are made of a phosphate group, glycerol group, and two fatty acid groups. You'll see the double bond in the other fatty acid that gives it that kind of a kink. In terms of the phospholipid, it's really the phosphate and fatty acid that we're often illustrating on these diagrams where we're drawing them, and the bilayer coming from the fact that there's two of these layers. That was something that's spontaneously arranged. We remember phospholipid had a head end that was hydrophilic, attracted to water, and tail end that was hydrophobic, avoided water, which explained this spontaneous arrangement of these cell membranes. Often embedded into cell membranes, we end up finding also proteins, various different kinds of proteins with various different kinds of functions, whether they're channels helping to carry materials across the cell membranes or selecting what can get across this cell membrane, or they might be transporters that actively move materials opposite to the natural gradients. They can be responsible for recognizing other materials, other cells, whether they're allowed to come into the cell. They can work as receptors, as we often see when we talk about hormones, for example. They can also function such like enzymes or form junctions from one cell to another. Because cell membranes are often made as a mixture of the phospholipids and the the proteins and many other substances that are found in the cell wall. They're often described as being mosaic-like in structure. You know, mosaic is this piece of art made of from multiple little pieces. The fluid part in the description refers to the fact that these cell membranes are constantly, the materials within them also moving, so they're not stationary like these pictures seem to suggest. They're actually constantly moving about. So often cell membranes are discussed as being fluid mosaics. How cell membranes sometimes help us to gain more surface areas by having these various different kinds of folds of these cell wall, cell membranes, phospholipid layers, whether it's smaller microvilli or larger folds. They typically serve the purpose of increasing the surface area of the cell. The inside of the cell is filled with cytoplasm. Cytoplasm is really all the material within the cell, excluding the cell nucleus. It's kind of this jelly-like fluid, mainly made of water, cells, and so on. Kind of fills the cell, giving it its shape and holding things in place, providing a place for a lot of processes to happen. Another cell structure, nucleus. Nucleus stores the genetic material. It is a double membrane structure, and within the nucleus we can find chromatin, which is a mixture of protein, DNA, RNA. We find genes. Genes are sections of DNA that contain instruction for a particular protein. For example, I often describe genes to my students as if they're a recipe in a recipe book. You never make all the dishes from one recipe book all at once. Similarly, you never have all the genes of the DNA active at one time. You choose which one you need, and then you make that. In terms of the nucleus at this double membrane does have small openings known as nuclear pores for facilitating passage of materials in and out of the nucleus as needed. I think it's important that we are aware that DNA always stays within the nucleus. It never leaves the nucleus. You can consider DNA as almost like this book that's precious in the library in a special collections department. You can't check it out. You can go and read it there, but you can't take the actual DNA out from the nucleus. But you can definitely make a copy of the book. So mRNA or RNA in general would be a copy of the DNA that's then allowed to be taken out of the library, so allowed to be taken out of the nucleus of the cell. We use this RNA strand, this copy of the DNA, in combination of tRNA to make decode that to make the actual protein by combining amino acids one after another until this chain of amino acids can be folded into a protein. So that's how we make something useful out of the instructions. So this process of copying DNA into RNA is known as transcription, whereas translation referred when we take this RNA and convert it into an actually useful functional protein. That's translation. Mitochondria, for the longest of time, we classified them, described them as a powerhouse of the cell, but now we know that ATP is produced also in other parts of the cell, but largely still within the mitochondria. They do need oxygen. They produce carbon dioxide in that process of producing ATP from carbs. We end up finding these folds inside the mitochondria known as cristae. And interestingly, mitochondria also have their own maternal DNA and ribosomes. Chloroplasts only found in plants and algae as a site of where photosynthesis happens, where we can convert solar energy, carbon dioxide, and water into sugars. And as a byproduct, we produce oxygen. The structure of chloroplast is shown there on the screen if you haven't had a chance to revise that earlier on. Endoplasmic reticulum is really continuation of the nucleus's wall, network of membranes And there's two types of endoplasmic reticulum. Endoplasmic reticulum that has a smooth surface, nothing attached to that. We describe that this is a smooth ER, whereas rough ER is characterized as having these ribosomes attached to the endoplasmic reticulum wall. These layers, these ribosomes are important in making proteins, as we saw. Golgi apparatus is a set of is a structure made of a series of membranes that have been folded, kind of wrapping each other. I often describe it to my students as a distribution center of a trucking company. So Golgi apparatus receives materials, sorts, reorganizes, repacks them, and then sends them out. So it kind of organizes materials that comes in and leave this structure. Vesicles, lysosomes, vacuoles all represent different kinds of structures that contain materials within the cell. They might be involved in moving those material around or storing materials within the cell. So that's to know about those. Centrioles are these pair of cylindrical tubes near the nucleus that are important in the cell division process as providing the cell spindle for the process of cell division. Cytoskeleton is really a network of microscopic filaments and tubules within the cytoplasm that provides kind of the scaffolding, the structure of the cell, but also you can consider it almost like providing highways where materials are able to travel within the cell as well. Obviously, we do know that there are certain differences between animal and plant cells. The big one with animal cells, that they have those plasma membranes that are not as rigid as the cellulose wall of the plant cells. Also, we end up finding in animal tissues extracellular matrix, which is important, whereas the plant cells really physically connect one to another most often. Cells are connected to other cells with various different kinds of junctions. Disjunctions are these more of zipper-like structures. Desmosomes, or also known as adhesion junctions, have filaments that connect from one cell to another and provide these anchoring spots for, especially for the filaments and other structures within the cytoplasm of the cell. Gap junctions are these channels from one cell to another, facilitating easy passage of material from one cell to another. So I think that covered all of the lecture material, kind of summarized, that I wanted to highlight. Only thing that I didn't talk a lot about or much about was the genes and DNA and inheritance patterns. I'll leave that for your own textbook to revisit. I was going to talk a little bit about the homework, but since it is just me here today, I'll probably save that for tomorrow's class. I don't think that there's urgency in us revisiting that. Like I said, I'm available for you in multiple ways, not only during these live online sessions, but also during the office hours. It's really important for me that I offer you chances for these discussions, for these check-ins. But if you are comfortable and your course is going well, I totally understand that there's no need for you to join if everything's going well. My role really is to be here to assist you, so do let me know how I can help you. And I'm going to jump out of this presentation. I just want to show one more thing, and that is me sharing the screen for our Canvas course. You should be seeing my screen now. And let's see if I can change it to a student view. So this week, you only have one learning module to work through. That learning module covers a revision of the themes of chemistry of life, cells, and genes, so chapters 2, 3, and 4 in your textbook. I provided some lecture videos, also the PowerPoints, and what I'm asking you to do is to make sure that you do quiz for chapter 2 and chapter 3 by the end of this week. Like I said, next week is going to be a little busier, so if you want to make already a head start of that, you are welcome to do so, but that's not required. Next week we will take our first exam. I did also receive a question from a student who found that it's easier for them to study cells through a diagram that they can color, so I prepared a nice little animal cell diagram. There's also a plant cell. You might label it, you might color it as you wish. I did provide also a colored version for you if that helps you to study. As always, if you tell me you need something, I will do my very best to facilitate your needs and to make sure that you have all the tools you need to be successful on these classes. Since it's been a little longer than I planned as a session, I'm gonna stop this session now, and I will be available tomorrow again for our live online session in case there's questions. The plan at the moment, unless there's something else that comes up, is that during that time we'll spend time on looking at the homework for this week. I appreciate you, and I hope you have a very good week. Let me know if I can help you. Otherwise, keep doing the great work that you're doing on this class. Until next time, I'll see you then.